Okay, so today we're going to tell you about the IIDP's Research Data Repository. The Integrated Islet Distribution Program is funded by NIDDK, and our mission is to distribute high-quality human islets, linked biospecimens, and integrated data to the worldwide diabetes research community to help advance basic science. We have been funded as the coordinating center for the IIDP since 2002. Here's the organizational chart. Dr. Abrams is our project scientist and Dr. Eggman, our program officer. I'm co-PI now with Dr. Evans Molina from the University of Indiana, an islet biologist and physician. James is our program manager. We have two initiatives I'll tell you about today, the Human Islet Genotyping Initiative, led by Dr. Gloin at Stanford, and the Human Islet Phenotyping Program, led by Dr. Bersova at Vanderbilt. We currently have 10 islet isolation centers around the country. I'll show you a map of those in a little bit. So as you all know, diabetes is highly heterogeneous and involves many organs and biological pathways. And although human islets have been available now for a couple decades, there's still much to learn about these pathways and the disease pathology and process and the cell makeup, et cetera. So it's critical to continue to get these islets out to the researchers. We also all know that diabetes is a growing problem around the world. Here's the CDC data showing that even undiagnosed diabetes is a major difficulty along with the total rate of diabetes. And if you look at some worldwide statistics, it's estimated that there's 537 million adults with diabetes today, 10.5% of the population, and it could rise to as high as 783 million by 2045, 12%. So it really is a major issue that continues to need study. Now, organ donation is critical to the whole IIDP and this whole process. And did you know one person, if they donate their organs, they may save up to 8 individuals by organ transplant. However, on the research side, it's very important too, and a single donation of a pancreas in order to isolate the islets, and then look at the research could possibly save millions of lives by donation for medical research. So this is a very crucial process, and we really appreciate all the donors who do give their organs to science. Now, the human pancreas is very complex, as are the islet cells, a different composition, histology, and makeup, and function. So this needs a great deal of study as well. Now, why don't we use mouse islets instead? It's been shown that although a lot can be learned, it's valuable to study mouse islets. They're not the same as human islets. And so we do need the donations from the humans. The architecture is different, the cell makeup, the response to cytotoxic factors, et cetera, differ among the humans as well. So here's the history of founding IIDP at City of Hope. Again, we began in 2002. At that time, we were called the Islet Cell Resource. The different color codes shows our funders. At that time, we were funded by the National Center for Research Resources. Then the JDRF, which is now changed to be Beyond T1D, started funding us for their investigators in 2008. And we were a clinical islet transplant program for a few years where we provided human islets for transplantation. But now we're focused fully on basic science as the Integrated Islet Distribution Program. We use that term because it combined the old ICR, the JDRF program, and the CIT program into one integrated initiative. And we're the largest provider of islets in the US today, and we provide them globally as well. The stars indicate that we've been renewed in 2012, then again in 2017, most recently in 2021 by the NIDDK. So we're funded through 2026. And as a landmark here, the data repository we're going to tell you about, the research data repository, has data going back to 2010. And that's over 1,500 records. We'll show you that. Now, we also have many partnerships I just want to mention with other diabetes initiatives, the HERN, for which we serve as the coordinating center as well, and their human pancreas analysis program and their PancDB. We have integration with them. A new program we will help to facilitate is PancBase, a knowledge database in diabetes. We have links and integration with pancreas for high resolution imaging. And of course, the dkNET has been our partner for many years, and now we are moving some HERN resources to partner with them, and the RRID, the research IDs are through their pathways, so we'll talk about that. So how does it work? So here's how the flow of islet distribution occurs worldwide. Deceased donor and their family provide consent to give the organs for the family provides consent for the organs, and those organ procurement organizations provide them to our isolation centers around the country. And currently, we have 10, as I mentioned. Now they isolate the islets, and then within 24 to 48 hours, we make a match between the offer that they have for an isolation and the researchers awaiting them. And we get them out using a standardized shipping protocol we've developed and send the tissues around the world. Then data are collected through the United Network for Organ Sharing. There's many variables they collect on the medical history, lab results, et cetera. Our donor centers also, our isolation centers also have donor data as well. And then we gather phenotyping data through the HIP, the Human Islet Phenotyping Program at Vanderbilt. A sample of the islets that are sent out to the investigators goes to the HIP so that they have standard pipeline and phenotyping on each of the isolations, and you can make comparisons across them for your experiments and others. And then the Stanford Human Islet Genomics Initiative provides genetic risk scores for type 1 and type 2 diabetes, as well as ancestry data. And then all of this is integrated into the research data repository that James will demonstrate for you in a few moments. Now, islet isolation, as we know, is a costly and logistically challenging process. And so it's taken us many years to assemble the right set of experts who can handle this whole process. And currently we have 10. They're located in Southern California at City of Hope and Proto Labs, Dr. Pappas at University of Arizona. We have Wisconsin, Ohio, Pennsylvania, New York, Virginia, and Miami, all represented by these expert islet isolation centers. And we really appreciate all the work that they do. We've found over the years it's varied from 14 down to 5, now back up to 10 centers. We feel that the number and diversity of centers helps us to ensure that there are sufficient OPOs and organs and sufficient islets generated with enough genetic diversity. The Human Islet Phenotyping Program, James will show you the data generated by Dr. Bersova in her lab, but they provide post-shipment phenotypic analysis of each isolation. And they include purity, viability, potency assessments. They look at the hormone content, histological analysis, the composition of alpha, beta, and beta cells, et cetera. So it provides a very rich analysis, both functionally and histologically, to go along with each isolation. The HIGI, the Islet Genotyping Initiative at Stanford, provides these partition genetic risk scores, now for T1D and T2D, excuse me, and visualization of genetic ancestry as well. So you can see the complete ancestry makeup of each donor, and if that's important for your analysis. The available IIDP resources are shown here. Not only do we have islets for non-diabetic and T2D donors, we have other biospecimens as well that go along with the islets, histology slides that are stained for each one, and the research data that we're talking about today. So that merges the UNOS, the IIC data, HIP, and HIGI. Our islet distributions have grown over the years. We've now done close to 300 million islets shipped through about a week ago, with over 7 million islets being isolated and distributed each year. These are very high quality islets, which is critical to the research. About 3/4 of them rated greater than 7 on a scale of 10 in terms of their quality, and only a very small percentage ever have a poor rating. And contamination is very low and contained as much as possible. Now, an important feature of our program that we developed many years ago, and there's some articles listed below, is how do we get these islets distributed in customized, equitable fashion. So we created a profile for each subscriber for the islets. They can put in their demand level. Is it high at a given time, standard, or low, depending on their research needs? They can define the preferences for donors that they would like to receive. And then we made a matching algorithm for islet distribution to optimize the distribution and broadcast to the awaiting investigators when an islet offer has been matched to them. We send web, phone, text to let them know it's waiting. The process for MAID basically is that we screen all the people who are requesting islets and are waiting to see if a given offer meets their minimum requirement. If it does, then we rank those who are waiting based on how long they've been waiting, how well it matches, those types of factors. We select an optimum combination of investigators and offer them the islets. If they accept, that's great. They're shipped out within 24 to 48 hours. If they reject the offer or there's no response, then we go to the next highest ranking set of investigators. Then finally, if time to get these out is running low and not all islets are accepted, we then broadcast what's called an open offer to a large group of investigators and hopefully place as many of the islets as we can in this manner. Now, here's the impact that IIDP has had on diabetes research since starting our distribution. There's now been over 2,200 isolations distributed to get out those 291 plus million islets. And we're supporting 594 studies, which we've traced to at least 955 publications that have used IIDP resources. And here's the global map of the 594 studies showing that we reach 16 different countries currently around the globe. Then the areas of research people sign up for is quite distinct. We have beta cell stimulus, death and dysfunction of beta cells, type 1 diabetes, alpha cell, genetics, genomics, engineering, transplantation, and we track all the different areas that people are studying. Now, the Research Data Repository, which is our main topic for today, has currently 1,546 donors. There are customizable filters up for these donors, so you can select by BMI, diabetes, sex, and many others. James will show you. And as I mentioned, it's fully integrated data from our centers, from UNOS, HIP, and HIGI. And it started in 2010 with over 200 data fields per isolation. And you can search, you can create a cohort, you can download the data, you can run customized reports or predefined reports. So far, 425 UNOS comprehensive reports have been downloaded. There's a publication checklist that now is required by many diabetes manuscripts that you can run on your isolations, and there are custom reports as well. So the benefits then, although we've had 2 decades of access to human islets, there's been a dramatic increase in understanding the human biology, but much remains to be known. So through IIDP, we have this standardized, transparent data on human islets, and this enables collaboration, reproducibility, and rigor and fairness. And there's an article that came out, an editorial by Hart and Powers a few years ago, saying that in spite of this, more needs to be done. There's not enough standard data and transparency so you can understand the quality and processing of each islet that was used in various research papers. And so they're encouraging more and more standards. And we had an editorial saying how we are answering that call for improved phenotyping and reporting of human islets. So some of the measures we've taken were the adoption of a standard identifier, the Research Resource Identifier, RID, through the NIH portal, so that there's a unique identifier for every islet portal preparation, and more centers are adopting this now. And then the RDR was released in 2019 to provide this integrated data along with the publication checklist that you can run for your publications that utilize IIDP islets. Here's an article that came out recently. There's a consortium called Bridge to AI. We all know that artificial intelligence is going to be changing our world. And this was a nice figure from that article about how to be AI ready. We feel that IIDP is striving for and achieving some of these characteristics. We have standardized data formats, seamless API integration, and protocol exchange. We register our bio in the NCBI biosample database for persistence. We have an ethics committee and data use agreement for ethical management of the data, a detailed data dictionary. We have the UNOS standards, harmonization, and quality assurance checks for characterizing the data, the RRIDs that were created in 2018, but we went back and backfilled all the way to the beginning, and then the RDR so that we promote fairness and accessibility and reusability of data. So we would continue, though, to evolve, and we want your feedback as you use the RDR. How can we improve even further and with further AI readiness and more features for supporting your research? Here's how the RRID is generated. We've created an automated module so that as soon as the Islet Isolation Center enters a few data points on the donor, a submission package is created at City of Hope, sent to the NCBI, and they issue the RRID. We check for that submission and response, and then we associate the RRID with the isolation record, and those are now accessible through the dkNET platforms as well. And then here's some examples of how RRIDs are showing up in the literature. Here was one article in JCI Insight not too long ago, where you can see that RRIDs are beginning to be in practice. They start with this SAM characters at the beginning, but not all of them have RRIDs. And if we can get this to be more ubiquitous, there'll be better traceability across studies. You can compare if you've used the same islets from the same isolation. So here's an article from Diabetologia where they did utilize the RRID, and this is from our publication checklist. This was the supplemental checklist submitted with this article, so that there will be more traceability across all the isolations. So let's get into the RRID a bit more. We'll do a demo. What can you study with the RRID? Well, we have cause of death, medications that were administered during the last hospital stay, laboratory results, the alpha and delta and beta cell breakdown, the medical history, genetic risk, and ancestry. So there's a lot that you can do with your hypotheses. Here's some potential analysis examples just to stimulate your ideas. You could look at the insulin curves by donor race and ethnicity and whether they vary, the insulin take-up. You can look at the genetic risk scores and compare them across donors. You could look at the percentage of beta cells by donor race ethnicity. There's also been studies, Dr. Brasova published this in a poster at the ADA in 2022, looking at islet structure-function relationship in this large pool of donors and how variable it is in composition among the three major islet endocrine cell types. She showed that islet composition varies across cell populations and across the sex of the donor and by beta cell composition and insulin uptake. So lots of interesting ideas that you could explore as well. We've done some research ourselves showing, for example, that the culture time influences the viability of the islets, and looking at multi-centered analysis of factors that might impact the islet yield, how many islets you actually get. Here's one table from that paper showing, for example, that the number of donor medications received during the last hospital stay had a significant influence negatively on the islet yield. And if you look at this paper, there were many other factors such as BMI, pancreas weight, et cetera. And here's examples of approved studies that are already being conducted using the RDR. One is on ancestry-specific modifiers of T1D. Donors of senescent beta cells is another project. We're looking at islet cell composition by donor characteristics, and your RDR study could go here. And I'll talk to you in a moment after the demo. I want to tell you about an initiative that we have for you to apply for an award up to $10,000 to fund your research in the RDR. That will be coming after this demo of the research data repository that James Cravens, our program manager, will now give you. So James, take it away and please show us the RDR.
Okay, thank you, Dr. Niland. Let me share my screen. So I will walk us through the data that we have available and how you can access those data. Is everybody able to see my screen?
Yes.
Okay. So that journey to obtaining our data starts here on our public website. This is iidp.coh.org. As Dr. Niland mentioned here, let me back up on this banner. We do have this funding opportunity right now that it's worth announcing, and applications are due in February. That will be for utilizing the data here that we're about to introduce you to. And one of the, of course, we want to utilize the value of the data here, but a objective of this program is to obtain feedback from all of you that are using our data. We developed this data portal really as a support system for our tissue recipients, the people getting islets and other tissues from IIDP. This was an easy way for them to download their data and make use of it in their projects. But now, as we've developed more data through HIGI and HIP, this data system is becoming more and more relevant on its own, and we really want to continue to expand this and improve it so that it better meets the needs of the research community. So any feedback that we can get from all of you would be great. Okay, so from our homepage, if you want to learn more about our RDR, if you go under Resources Offered and then Research Data Repository, this brings up a summary of all the data points in our system. You can see, as Dr. Niland mentioned earlier, we have over 1,500 isolations represented in the RDR. There's some visuals here that kind of show you, if you mouse over, how many of these records are represented by these different donor demographics, the age, the sex, the donor race, BMI, diabetes status, and then some of the resources that we have available. We have histology slides available for 604 of our 1,500 plus records. We have some frozen islets available as well, 73 samples. And then we have phenotypic data from our HIP program and genomic data from our HIGI program as well. And so we have been seeing roughly, about 75 new isolations per year over the course of the last 10 years or so. Things slowed a little bit during the pandemic. So if you do join to access our data, we probably see somewhere between 5 and 10 new records each month. So it's worth coming back from time to time. You'll see that new data are added. And then so one important distinction I should make, this view right here on our public website, this will show you all of the data points that we have. I'll scroll through this list with you in a moment. But to actually access and download the data, you will have to click this green button in the upper right to apply. This is based on requirements from City of Hope, who hosts our data, as well as UNOS, who provides us with a ton of really rich donor data. Both require that we house our data on a secured server. So to download the data, you will have to submit this simple form. It's really just a registration process, and that will gain you access to our data. So what data do we have? Really, it falls into two main categories: data about the donor, and then data about the islet isolation itself and the islets themselves. In parentheses, you can see how many of the records will have these various data points. So donor information is available on all of the records, but then breaking it down further, there might be some fields that are missing on some donors, but we have basic demographics. We have a lot of clinical information. I should have also noted that our donor data, some comes from our centers when they broadcast new records through our program, but the majority comes through UNOS. And then we also get data, of course, from HIP and HIGI. So, and here's a good example. So HIGI, the genetic information, this comes from our Dr. Goins' lab at Stanford. So these data points are available. You can see just under 400 records, and we're expecting a new group of data to be deposited early in 2025. So that should increase significantly. Serology information on the donors. I'll scroll through it kind of quickly. Cause of death, diabetes history, and some of the complications and family history of diabetes, therapy for type 2 diabetes, Oh, good point to make here too. So we have, out of the 1,547 isolations represented in our data set, our database, 119 had type 2 diabetes, and then over 1,400 were non-diabetic. We do not offer islets or other tissues from type 1 donors, so our data reflects that as well. We have data from type 2s and non-diabetics, but not from type 1. So social history, this comes from UNOS, cigarette use, alcohol use, drug use, different medications, which has been curated very carefully by some of our staff, and we had a med student help us to really dig into that and standardize all of this data, organ recovery. So isolation data, what data do we have? Basic information about the procedure and the organ acquisition, culture time, things like that. Islet characteristics, we have the islet index and grade, the purity viability, the glucose stimulated insulin release stimulation index provided by the centers prior to shipping the islets, sterility data, of course, and then our phenotyping data from HIP. So that includes the purity viability, functional analyses by parafusion, the insulin and glucagon responses, hormone content, and then the histological analysis, the cell composition and tissue composition. So all of those data points are available. If you see anything here that hopefully that piques your interest and you have some great ideas, it's worth looking into that DRTSA opportunity. And that would be a separate application process. But if you are interested in obtaining the data but not applying for that award, the journey really starts here, this green button to apply for data. Once that simple application form has been completed and approved, you'll have access to our system. So I'll jump in now. This is our test system. So some of the numbers might vary slightly from what I just showed you, but it's the same basic setup. So once inside, you'll see your study listed, and then all of the data are accessible here through our RDR portal. So when you go in here, so here's the RDR. It's really two main components on the screen. We have on the left side these various filters. I'll expand all these out so you can see them. The filters will help you to narrow down and customize this list here in the center of the screen of all of the records. As you're filtering and using these filters, you'll see that this number here on the left will change in real time as a smaller subpopulation becomes visible. It's a three-step process. Once you've gone through the data here and made the selections of the records that are of interest to you, the second step would be to define the data points that you'd like to view. We do have some data here, of course, that you can see that help you to kind of identify if it's a record that would work for your research, but all of the vast data that we went through in that data tree, that will be available to you in step 2. And then, of course, step 3 is to view the final report and download it if you would like. For any of these records that come up, you can always click on any of these links and it'll bring you some visualizations. So let me utilize some of these filters here and you can see how it works. Again, this system was really set up for our tissue recipients, so we kind of have a model that's sort of islet isolation specific. So you could break things down by the most pure fraction or the less pure fraction, donor age, the sex of the donor, the BMI, diabetes status. I guess for now I'll click on non-diabetic, ethnicity, race, the type 1 overall genetic risk score, the type 2 risk score, and then the resources available. So if you are interested, of course, in obtaining frozen islets or slides, this is a great way for you to see which records are available. You can, here, let me show some of the detailed data. So I'll click into records that have both genomic data and phenotypic data. So now I have a subset, as you can see, it kind of filtered here to 297 records that have all of those things. They're from the most pure fraction, they're non-diabetic, and they have genomic and phenotypic data. We do have a nice feature here. if you intend to come back, as I mentioned, we do see several new records every month. If you have set a series of filters that make sense for the project you're doing, and you check back, you can save this customized filter list, and then that will be available to you when you return next time. So for now, I'll just call this test 1. I'll reset all this. So if you were to come in, let's say next month, you'll see here you now have a saved filter. If you come down from this drop down, test 1, and then it sort of resurrects the filters that you had defined earlier. So it makes it easy to just jump in where you left off and see if there's been any additions. Similarly, if the records themselves are what you're interested in, like let's say you're working on a specific analysis involving, well, in this case, 297 donors, and these are all of interest to a particular study, you can save just this list, this cohort of records, and the same thing, you can, test cohort, I'll call it 1. And then the next time you come in, you can pull that up from, let me reset everything. So test cohort 1 is saved, and then those 297 donors will come up. And then, yeah, if UNOS data are updated or if additional HIP or Higgy data are entered into the system, then now you can easily recall all of that specific cohort that you were looking at. Okay, so this, as I mentioned, is just a brief overview. You can see some of the demographics and things and the risk scores and whether or not certain resources are available, but maybe you want to dig in a little bit further before settling on these records as the ones that are important to your research. So if we click on one of these links for the RRID, it'll bring up the visualizations of the data. It takes a second to load. So we'll have all the data that our centers made available when they broadcasted the tissues originally, as well as the data from HIP and Higgy will be viewable here. So here's just the basic overall data on the demographics of the donor, a sneak preview of some of the HIP and Higgy data, which as I scroll down, you'll see some more details. Here's the genetic risk scores, kind of bell curves, and then the broadcast data on the donor. So basic demographics, which other RRIDs might be relevant if other tissues were also broadcasted from that same donor. For those of you that don't know, the RRID is tissue specific. So we make it convenient to link up other tissues from that same donor, what RRIDs were associated with that same donor. The sterility results, you can scroll through all this, make sure this is something that is suitable for your project. Okay, now we're down here in the HIP category. The color has changed. So there's a lot of really nice images from HIP that are uploaded here. And if you click on any of them, you can see, let me make this a little bigger. You can see it and zoom in, and then you can even download a full image. What else? Okay, so here's all the parafusion results. So this kind of interactive traces of the parafusion, both the insulin and the glucagon responses, 2 different units. Stimulation index, the area under the curve. And then some of these images here are really nice, and we have links to Pancreatlas, where you can get really nice high definition images. And that's, I'm pretty sure, I think that's publicly available, but similarly, I think you register for an account and then you can access all of those high definition images through Pancreatlas. Okay, Higgy data. So here's the genetic ancestry data visualized in this donut chart. And then also the table here. If you expand the table, you'll see all the different genetic ancestry components. One thing I should have mentioned up here at HIP, if you have any questions about the protocols and how we are generating the data, these little kind of green icons here will provide you a link to the protocol so you can find out more information about those. Down here in Higgy, we can see also some links to some of the background information of the protocols that are being used, as well as a visual of the pipeline and how the genetic data are produced. Here are the type 1 genetic risk scores, and then the composition of the risk, HLA versus other genetic risks. Again, there's some helpful links down here for more information. The type 2 risk, same thing. And then at the very end of this visualization pop-up, it has all of the images that we have for that prep. So some are the pre-shipment images that come from our islet isolation centers, and then the others are the post-shipment images that come from HIP, some of which are available in pancreas. Okay, so back to the RDR itself. So now I have my test cohort 1 here. Let's say I want to download data on all of these. I can select all of them. If there's only specific subsets that I want, of course, I can deselect and make a custom choice. Step 2. Okay, now I'm here where I can define the data points that I'm interested in. We have 3 predefined reports. These are commonly requested series of data that we have set up for, one click of a button. The first being the comprehensive IIDP and UNOS data report, so all of the data that we have, if you just need all of the data. The second one, which we're really trying to encourage the use of, the Islet data checklist for publication. So this, as Dr. Niland alluded to earlier, some journals now are requiring the authors to provide a standardized list of data points to define the donors, that adds to the transparency of the project, and also shows, encourages, I guess, reproducibility, where you can verify what was done. So that we have that template set up already based on that Hart Powers paper that was published in 2019. And then we have a phenotyping data report that shows all of the data from our HIP program. To be continued, we'll add in the near future a genotyping report as well. So all of the data from our HIGI program will be a one-click report. Okay, now if you wanted to do instead a custom report, you could click on that button, and then now the same data tree that we went through earlier is available, and you can click on either everything, maybe all donor information, maybe just the demographics, or maybe you have individual data points that you'd like to include. Anywhere you see one of these triangle caret, you can expand and then click down and make your selections, and continue through the data tree with your selections. You could also search for a data point if there's something specific that you want. And that's that. For now, though, let me just demo the checklist for publication, since we're really trying to encourage that. So for all the 297 records in that cohort that I defined earlier, we will now create this checklist for publication report. That's step 3. It generates the data in real time, so every day you'll have new data here, or updated data. You could, of course, scroll through and browse the data if that's all you're interested in. But you could, of course, let me scroll down to the bottom here. You could also download the report in Excel or in CSV. So if I click on Excel, it'll download, and in a moment I will pull it up, and you will see it also really closely mimics those examples that Dr. Niland went through earlier with some of the papers recently that did utilize one of these checklists. So, is it loading? Here it is. Okay. So yeah, for those of you that aren't familiar with this paper that was published by Hart Powers in 2019, I think, these were the key data points that were recommended as mandatory to kind of define the donor itself. And there were also a few other donor data points that were recommended that would be beneficial, but maybe aren't as essential as those mandatory points. So for all of the 297 records now that I just included in this report, we have this this checklist ready to rock, and you print it out and submit it to the journal that you are trying to publish in. So that is that. I think that is all I have to say about the RDR. I don't know, Dr. Niland, was there anything that I glanced over, anything that I should focus on before we finish things up?
I think that's great, James. And we can take questions at the end if anybody would like to delve into another feature. Great, thank you very much.
Okay.
So I will just share my screen again for just a few last slides. Okay, so Just to tell you a little bit about what do we plan in the future. IIDP never sits still. We're always trying to evolve and evolve the data and the resources. So we have some novel scientific initiatives ongoing. We did a Technology Resource Award last year, and we're developing new quality control measures. We did a previous round of a Data Resource Trainee Scholarship Award. I'll tell you about the new one now. And we also periodically offer the Islet Award Initiative that gives free islets for research. So investigators can apply for that and get some free islets to do their experiments. And we're trying to always improve the offerings, such as human pancreatic slices, ductal cells, et cetera. We want to augment more the non-islet biospecimens that we can match with the islets, perhaps size-sorted hand-picked islets or more juvenile donors in future, and protocol and quality control improvements, and expanding the tissues offered based on surveying the investigator demand. And then we always want to increase the sharing and the usage of IIDP data. So we'll continue to expand the data available in the RDR and enhance its usage through awards such as the ones I'm going to mention in a moment. And then we hope to have additional IIDP supported publications using this data. And I have some hot off the press information at the end here regarding a new publication that will be coming out. So here's the funding opportunity you've probably been waiting to hear. There's no charge to use the RDR as it is. You can go in and, as James said, apply and just give us a brief synopsis of your research, and away you go and use the data. But if you would like to apply for this competition, become your own PI, be able to brag about your scholarship award here, we are offering this $10,000 opportunity to utilize the RDR. So the goal is to help advance human pancreas biology and research by providing trainees with support and access to detailed data that you've seen today in the RDR, so you can conduct your own secondary analyses of the relationships among donor characteristics, phenotyping, and genetic risk. Postdoctoral fellows, graduate, and medical students are eligible, including at the master's level or PhD, MD. There's one caveat that you need to have a mentor designated who will help supervise the activities and be familiar with the research and commit to the career development of the awardee. And the deliverables, your responsibility will be on conclusion of the project to provide us with a final progress report that summarizes your analyses, your results, what impact you feel they may have on diabetes research, and importantly, as James mentioned before, give us feedback on the design and functionality of the RDR so we can continually improve the future functionality, utility, the types of data included, et cetera. So that's the opportunity. It's, as I said, $10,000 total. That would include any indirect costs that your institution may have. That percentage varies. You can ask your grant office. Here's the link down below as to how you would apply. You can also go to our website and iidp.coh.org, and the announcement will be there. Here's some key dates. We posted it just a few days ago, so it's hot off the press. There'll be an informational webinar on December 6th. If you'd like more information after reading the RFP, we can answer your questions about the application process, et cetera. A letter of intent is always nice if you plan to apply, so we know how many reviewers to line up. That would be due December 16th. As mentioned, Valentine's Day will be the application due date. And then there'll be 6 to 8 weeks where we engage scientific reviewers to take a look at your application, announce the awardees in April, and your start date would be in May of next year. It can go up to 2 years of it. Hopefully it wouldn't take that long, but you'd have up to 2 years to utilize the funds and conduct your research. So we're happy to answer any questions about that in a moment. And then I just want to finish with some acknowledgments. Thank you so much to our whole team, many of whom are on the call today. We have an excellent set of developers, scientific experts, and program managers. Thank you to our 10 islet isolation centers around the country who are providing these precious islets. I appreciate my co-investigator, co-PI, Carmela Evans-Molina, and Dr. Brasova at Vanderbilt, Dr. Gloin at Stanford, leading our initiatives for phenotyping and genotyping. Thank you to the NIDDK for funding us these many years, and Breakthrough T1D, formerly JDRF. And of course, very importantly, most importantly, thank you to the people with diabetes and the organ donors and their families. If you want to contact us, there's many ways. You can follow us on X, although I think we're going to be moving over to Blue Skies soon as well. IIDP.coh.org is our website. You can email us, and we run a HERN newsletter that goes out every month and would have this award announced. It would have other awards. It would have job opportunities. So sign up for the HERN newsletter if you'd like to stay abreast of recent scientific achievements and HERN accomplishments as well. And then this is like hot on the press. There is a new article that Dr. Bersilver's spearheaded with Dr. Evans Molina that is now in preprint on BioArcs IV called, I didn't even have time to put the title in there. It's called Heterogeneous Endocrine Cell Composition Defines Human Islet Functional Phenotypes. It'd be very interesting to show how you can utilize this type of data. And there's the QR code if you want to go to it as well. So thank you very much. And Ko-Wei, we're happy to take any questions.
Yeah, thank you, Joyce, and thank you, James, for the fantastic presentation. So now it's our Q&A session. So if you have question, you can unmute yourself and ask question during this time or continue to use chat function. So there is one question from the audience in the chat. So Anita from the RID project, it is wonderful to see the RIDs in many, many places, but I wonder if we can move the authors a little more to make sure to include this in all of their publications. I wonder what things we can do to help get this more accepted in relevant journals. For the publication checklist, is it possible to add the letters already in front of those same numbers so that our pipeline find the paper?
Thank you, Anita. That's a great point. Yeah, we should do that. I kind of noticed that when I was showing the two different journals and how they were presented. So we should add RID. We do need to continually get the word out. One initiative that I know of that may help with this, the Pank Base initiative of HERN that I mentioned. It's a newly funded knowledge base that's going to hopefully integrate and harmonize across many diabetes data resources. And I know that Kyle Galton at UC San Diego and Anna Gloin at Stanford and interacting with us are already looking at papers and trying to figure out where was the RID used, where was it not. If it was not, can we, help to identify the samples used and try to go back to the papers and encourage them to include the RID. So that's one initiative. But yeah, other ideas are welcome. And I know that we also should probably do a publication directly on the RID process and this automated module that our group has.
I think that's been in the works for some time.
Yeah,
But it's always great to get those papers out.
This is a good spur
but
on.
Actually, if you do have somebody working on that, do you think you could get them on a joint Zoom call with us so that because we have a lot of our students go through this. We have a couple of automated pipelines that kind of go through this data, and if we're not finding those papers, then maybe we need to tweak something.
That's a good idea. Yeah, let's hook you guys up with them and we can have a good call.
That would be lovely, because we think about this stuff all the time, and we have pretty good help from the endocrinology journals. They do require RIDs for at least antibodies, but I'd like to push them a little bit more to see if we can get a requirement on these tissues, or at least a reminder, or for the authors, at least of endocrinology and Journal of the Endocrine Society,
yes,
to get those those requirements in front of the authors in a little bit more transparent way. So I'd love to check back in with you guys and see if we can get a little bit more movement on this. I know we've had some papers, but it's not quite enough. You guys have sent out tons and tons of these islets, and we're not getting back the percentage of papers that we would think that would be commensurate with that at this point.
I agree. And we did a search recently, our librarian did. The only journals that we can find that are mandating this checklist with RIDs and metadata on the isolations are Diabetes, Diabetologia, and Diabetic Medicine. Maybe we can also approach it at the journal level. Possibly we could approach other journals and see if the mandate could be extended.
I think that would be great. And again, updating that checklist to have the RID in front of those salmon numbers
Mhm.
would be lovely, because then we would at least find the paper based on that, having the checklist if somebody is including them.
Yeah.
If not, there are lots of salmon numbers from lots of different unrelated projects. We don't routinely search for those.
Great idea. We will add that. Thank you, Anita.
Thanks.
Marcella, did you have a comment or question?
Yeah, I would just like to follow up on comment from Anita. I think motivating journals to document RIDs for human islets is going to be really important. And I think it's going to be up to us scientists reaching out to journals and editors of journals to implement this really important information in every publication that reports on human islets. I'm part of pancreas knowledge base team, and I fully understand how difficult it is to track data, other types of data on human islets without RIDs. So I'm in your camp, and I would love to make it happen with the journals.
That's great. Thank you, Marcella. We're going to enlist that help. We appreciate it. And the editorial that you helped spearhead, answering the call for this was great.
Okay, so there is another question from Dr. Jordan, right? Is pancreas weight data available? And for the histology slides, is it recorded what region of the pancreas, for example, head, body, tail, they come from?
Good questions. James, do you want to field those?
Yeah, so I did just check in that data tree right now, and I did not see pancreas weight, and we don't ask our centers for that. I do see that our database administrator, Jenny, is here. Jenny, do you happen to know if UNOS collects that, if that's something that is obtainable to us if we were to expand our request from UNOS? I'm not sure if she's still on the call, but
I just looked through it, and I didn't see it on the UNOS website export list, but I can reach out to them to see if they can make it available to us.
Okay, thank you. So yeah, Jordan, if I remember correctly, I think you are in our database already as an IIDP user. I can reach out to you when we find the answer to that. To the second part, we don't have the centers specify part of the pancreas, but our protocol calls for the sample to be taken, I believe, from the tail. So again, I'll confirm that and I'll reach out to you with the answer, but it should be coming from the same part for all of the different samples, all of the different slides. So.
Great, thank you, Jim.
Okay, thank you. And there is a question from the chat sending to a direct message. So what information is required to apply for data access? Do I need to submit a description or proposal or prepare any specific document?
It's fairly simple. James, you can maybe elaborate more, but when you go into that green button that he showed you, apply for RDR access, mainly it's your name, position, your title, which can be anything, but and then a brief abstract about your research. It's not very long. It doesn't allow you to put in too much. I don't think there's any other documents required for RDR access like BioSketch or anything. James, do you want to elaborate?
Yeah, I don't think so either. It's been a while since I've ran through that as a test, but yeah, it's really simple, and everybody is basically approved as long as you've completed the application completely. So it should be an easy process. I do want to reiterate, though, that for those of you that are interested in the DRTSA award, that would be a separate application process. So follow the, and maybe Dr. Niland, since you're still sharing your screen, if you can go back to that link for the application. So yeah, there it is. So that link is, or even just going to our homepage at iidp.coh.org, that will show you how to apply for the DRTSA. It's a limited group of, it's postdoc fellows and some grad students, but we have received some feedback this week, interested researchers that are also early stage researchers, but more on the PI track. And we are going to, hopefully in the next couple of weeks, get a sort of parallel opportunity out there that's a little broader for those people as well. So stay tuned. There might be another funding opportunity. But yeah, those would be a separate application process. Anybody who just needs a standard data study from the IIDP, regardless of this, independently of this funding opportunity, that green button will take you through the process, and it's just a few quick questions.
Okay, I think he said thank you. And how long does this typically to get approved? Can international investigator apply for data access?
So standard data study, yes, everybody, it's open to anybody, and it's a very quick process. Generally, it's approved and access is granted within a week or so. The DRTSA will be a little bit longer, and that is it's a U.S. citizenship. A U.S. citizenship is not required for the DRTSA, but if somebody is a non-citizen, they do have to be located at an institution in the United States to be eligible for this funding opportunity.
But the RDR is open globally. It just needs the same approval process, and it only takes a few weeks to get a turnaround on approval.